Okay, hello. Hi, everyone. We want to present today our presentation about our project, which is an IMMU VIS towards the pan marker and symmetry invariant tissue embeddings. So before jumping into the project, let's discuss and let's describe the image, that data set that we are working with. So basically, what we are having is a data set that is kind of unstructured in terms of the selection of the markers. And before proceeding with the experiments, we want to select markers, which then are proceed by the creating tissues and cutting the smaller chunks of it, which are then sent to the special machine, which is called cytometry. And this one then is using the special metal tagged antibodies, and the laser is punctuating the slices and creating the kind of spectrum of this tissue, which create this data set that is up to several markers. For instance, here we have 40s, and with that, then we are able to create experiments like clustering, like phenotyping, and so on.
So, problem statement. What we are supposed to do, our task basically is to create foundational model for multi-marker IMC medical data. These data sets are kind of challenging because these images have high resolution. They are large and they are quite noisy because they consist of large amounts of cells. So like the images have like important information at a very narrow, or like each patch of image has a meaning. So we have to take a look like at the entire image. And also different data sets can have different subsets of the markers. So we train the model on one data set, and it should also work on the subset of markers instead of the entire set. And also the images itself can have irregular shapes. So 600 by 600 is one of them, but it can vary.
So in our work, in my work, I've been utilizing recent advancements in group equivariant convolutional neural networks. So GCNNs and steerable CNNs. Tomek and I have been utilizing SimCLR for making our models robust for rotations, and Tomek also uses PigVire to produce rotation equivariant global representations of whole slides. And Mateusz is working on utilizing VITMA, so training vision transformers in mask autoencoding setting for obtaining representation of patches.
As Szymon already said, the one part of our project is to create a multi-marker patch embedding model. So the idea is that first, at a very small patch level, we train a tokenizer using VQVAE with slight modifications for small patches, like 8 by 8 pixels. And it's marker agnostic, so one token corresponds to one marker. One image has 40 markers, then there are 40 tokens for the patch. And then the second part is to pre-train a visual transformer via massive patch prediction on grids of these tokens. So then we are dealing with larger images like 100 by 100 or 64 by 64. And then the visual transformer encoder could be used as a foundational model for a variety of tasks like classification. So our modified VQVAE, normal VQVAE has just one token for image patch. And here, because the lots of markers are very dark, but they still can have meaningful information, and the intensity of the marker is also important. So we divide the token into three parts. The first one is the texture token, which is just normalized mean 0 SCD1. The second part is the patch mean token, and then we have a image STD token. Then the token for a patch is just a combination of these three tokens. So the second step is the visual transformer. On a very high level, Maya just works that we have an input image with some of the patches randomly masked, in our case 50% of them. Then the encoder encodes the non-masked patches, and then a smaller decoder, smaller because we would like to have a powerful encoder and then discard the decoder, is supposed to reconstruct the missing patches of the image. So basically the training, since we have marker agnostic VQVAE, we have, for example, 40 tokens per patch. Then we have for this marker embedding from the VIT codebook. Then basically to combine them, we concatenate the patches and perform a linear transformation to combine them. Then a standard VIT transformer training, Maya training with non-causal attention results in the pre-trained VIT.
So I've been developing a new method for foundational tissue and autoencoders that can utilize the knowledge of markers that we want to process. So how the simple version of such autoencoder might look like. We can first project each individual marker patches into a shared latent space with a linear projection or one-by-one convolution, and then sum those projections. Then we can operate on this shared latent space and feed it further into a regular autoencoder to obtain a feature space of lower spatial resolution, and then we can reconstruct individual patches with an inverse mapping that is also a one-by-one convolution. And how we can improve that? We could try ingesting the information about markers that we possess into the autoencoder, and we do that by dropping a superkernel block after each regular convolution block, and we wrap it in the residual connections. So the superkernel block works by producing on the fly kernel for the convolution to enrich the sample features with the information conditioned on the marker set that the sample is that the sample consists of. So first we further project the output of the convolution into a lower dimensionality space, and we apply the generated convolution on this lower dimensionality space, and then we project back into the space of the convolution block. So the superkernel channel chart, which generates the conditioned kernel, is a transformer encoder that is conditioned on marker embeddings and produces this dynamic kernel. Another method that I've been researching is group convolutions and steerable convolutions. The first method is are convolutions that are equivalent under discrete rotations and reflections, while steerable convolutions are equivalent under continuous rotations. So here we can see how the steerable convolutions can be visualized. In this setting, the feature space consists of two component activations that encode a magnitude and orientation of the length patterns. And we can see that the vector field is rotating with the angle that we rotate the input image. And another difference between steerable convolutions and regular convolutions is that we don't, that the kernel which we apply is given by the linear combination of length parameters, which we can see in the right bottom corner. Of those this linear combination of length parameters with pre-computed kernel bases, which are now to preserve equivalence on this feature field.
Yeah, and as the final module of our solution, we want to not only have the great embedding for the patches, but also one representation that is global. And we aim for the representation that is invariant for n transformations, which is the rotation of the 90 degrees and the symmetries. To do that, we use the autoencoder with the contrastive learning. And the architecture of this can look like this. So basically, we have a graph that's representing the image. We add the additional graph node that is actually is used for the CLS token as a kind of latent, from where we then can actually have the global representation. And to preserve the invariance and the learning permutation, we introduced this permuter model that classified or learned each of the 8 permutation, and then the task is to reconstruct the image graph as usual for the autoencoders. And now the experiment and the results.
Experiments for the multi-marker patch embedding model. So first, the mean NCD cluster visualization. Maybe we can just skip to the actual. Okay. So for the tokens, first of all, you can see that the patches have very skewed distribution of means and STDs. And also here you can see the quantized, the tokenized statistics. So we have mean and STD split into a few components. Per channel, it also varies significantly. Some channels have quite wide distributions, while others are very dark, basically. Okay. The same for STD. Here is the codebook of the texture codebook, the marker agnostic one. And here we can see some reconstructions of the grids via just PQVAE. And here we can see like the image patches reconstruction, the masked patch reconstruction using the VIT MAE. Also, I found a classification fine-tuning where I had like 67% accuracy on a very simple 4-class dataset, 67% on the balanced version of the dataset.
In my experiments, I've focused on measuring the impact of lack of rotational invariance for some previous work on imaging mass cytometry. And for example, the Sorin et al. Paper uses a simple ResNet model to produce flattened representations of every channel, which then are concatenated and used in analysis. So to emulate this setting, I trained some models, some vanilla models and models with contrastive learning objective to see how we can improve the rotational invariance. This is a Disney mapping of channel of patch embeddings of original patches and rotated patches, and we can see how they differ between each other. And in those experiments, I've measured the average cluster retrieval accuracy across different rotations and different number of clusters. So for the vanilla tissue convolutional embedder, we can see that the accuracy is dropping very fast. And for the version of the model with contrastive learning objective at, the accuracy is not dropping that much. I've also run experiments to measure effectiveness of the superkernel block. So it showcased that it has a positive impact on training, but I had some problems with converging on the test set, and further experiments might be needed on larger data sets to measure the impact of the superkernel block. And here are some examples of inputs and reconstructions given by the equivariant autoencoder. So here we can see on the left side original input and reconstructions on the bottom, and on the right side input rotated by 90 degrees and reconstructions that are almost perfectly rotated as well. And this applies also for other rotations like 45 degrees rotations.
And just last sentence. So for the last part, we also checked this invariance for those global representation and showed like how this looks like in the latent. And one disclaimer that because it's kind of a sequence model, the first initial experiments are for a MNIST model. So you can see that this is the reconstruction for the MNIST, but of course in the final model we will do this for the IMC data. And here are the predicted learn permutations. And here's the PCA of this global representation. We still try to force them to be close to each other because right now they are in groups of 4s. And the future work is to conduct more experiments and creating better this invariant representation. We are also aiming for publish our work in some scientific journal, maybe other testing other architectures and compare to other benchmarks or create even one for us. Thank you very much. Are there any questions?
I have a question.
Okay.
So regarding this VIS model, like with this code, have you thought about maybe were these like cherry pick or were they like because they look super cool?
Which one?
Maybe the three one, I think.
This one or that one? This is
Yeah.
from the VQVAE, this one is from the VIS.
Nice. So, like in terms of, so have you tested, for example, the uncertainty of these tokens, like whether the model is certain or uncertain in terms of masking? And
No, not yet. I mean, I think like what looks promising is like the quite small, I mean, cross entropy loss in general, which kind of measures the certainty, right? Like, yeah, but I think there's still a bit of work to be done to like make sure these ones are robust and like good reconstructions of like entire data sets, not just
yeah, because I imagine it might be hard to ask, but if they are as good, maybe it's not, there is no need for actually measuring any sort of uncertainty. Like, okay.
I think like like using this foundation model for further tasks maybe is a good like measure of the quality. Right?
Super cool. Thank you.
You.
Any questions?